Good afternoon. What we're going to do today is look over the replication of DNA, which is key area 2 of Unit 1. Now, DNA replication is something we have touched on before in Nat 5 when we looked at mitosis. As it says, DNA is a unique molecule in that it can reproduce itself exactly. So you can go from one DNA molecule to two DNA molecules, and that's the process we're going to be looking at. Now, there's one of the main molecules in this is an enzyme called DNA polymerase, and we're going to discuss that. The clue here that it's an enzyme is the letters ASE on the end. That's quite common when we name enzymes. Okay, if you think about lipase, protease, anything with A's tends to be an enzyme. So that's just a little hint there. Okay. Here we've got DNA polymerase needs a primer to start replication. That's something we're going to come to when we look at the stages, and we'll discuss that in more detail. Here we've got DNA replication is said to be semi-conservative. Each new molecule contains one new strand and one original strand. So if we have a look at the diagram here, you will see that we've got our original DNA molecule, which kind of looks like a ladder, okay, because it's been instead of the double helix twisting round, this has unwound. So now it's just straight up and down. And on this side, you've got one strand of the DNA, and on this side, you have the other. And they're joined together by their base pairs, complementary base pairs that have bound together with hydrogen bonds. Now, the first thing that happens is the two strands have to separate, okay? And then the third step here, you can see that each strand has a new strand added to it. So each new molecule has one original strand, which is in the red, and the blue represents a new strand. So each molecule has an original strand and a new strand. So you've gone from one DNA molecule to two. We're going to break it down further. So when does this all occur? Well, this occurs just before mitosis. Now, we looked at mitosis in Nat 5, but this is a process which would occur just before mitosis takes place. As it says, the enzyme DNA polymerase adds nucleotides according to their base pairing rule. So when we look at the steps for DNA replication, just bear in mind the rule that we've already established, that A, or adenine, always base pairs with T, thymine, and G always goes with C, guanine and cytosine. Right. So there are quite a few steps here, so just bear with us. So the first stage is the DNA molecule needs to unwind. So we know that DNA is in double helix. That means it's two strands twisted around each other. First thing that happens is they unwind so that they are just straight up and down like a ladder. Secondly, the weak hydrogen bonds between the base pairs break, allowing the two strands to separate or unzip. So if you have a look, at the diagram, these ones have not broken yet, so they are just as normal, but these ones, the hydrogen bonds, have broken between the bases, and you can see that they are now starting to unzip. And you can see that it is like a zip on a coat, okay? It's starting to come apart. Now, that's because the bonds that join these base pairs, hydrogen bonds, are broken. That's why it's important that they need to be weak, so that can happen. The third step, their bases are now exposed at a Y-shaped replication fork. It's called a Y-shaped replication fork because it looks like the letter Y, and these bases are now exposed, which is really important for the next stage.